Sound clear.
Hi, good morning. I hope we are keeping well and taking all precautions to avoid the pandemic. As you know, the number of cases have already gone up in the last 1 week or so. So we believe that there is a second resurgence of the infection. So we need to be careful. We cannot be lax about it. But today I'm going to take off the mask because there is nobody in the LT. It's online class, and secondly, the voice gets muffled once you have the mask. So please excuse me today. So we are going to talk about the bacterial genetics. Uh, Basically, I will give you an overview, and the overview will be uh, revisiting your class 12 class on the prokaryotic and eukaryotic differences, etc. But the whole idea of taking this seminar is that this will give you a basis of understanding how the knowledge of the bacterial genetics or molecular structure of the bacteria helps you to understand the disease, helps you to understand how the disease can spread. It also helps you how the organism avoids sometimes the immune response. It also helps you to understand how the organism transfers their antibiotic resistance from one bacteria to another, and it also helps to understand how you can utilize this knowledge of the bacterial genome to your advantage. So let's start. What do you what do I expect that you should know at the end of the lecture? This will help you to understand the basis of molecular diagnostic assays in infectious disease. Once you understand the basic of bacterial genetics, it will help you to understand how the nucleic acid based assays are carried out in the laboratory, how a specific gene is targeted, how one can utilize this information about the genome of a bacteria, whole sequence of the bacteria, and build up a polymerase chain reaction or PCR or a real time PCR. As you realize, with the corona pandemic, that real time PCR or RT-PCR is or for even for example for influenza virus infection, swine flu or the bird flu, avian flu. In those cases, only nucleic acid based assay or real-time PCR are the gold standard. So this will help you to understand how this how this methodology or how this molecular diagnostic test work. It will also help you to understand how drug resistance shift from one bacteria to another within a very short period of time, either through a jumping gene or some kind of a bacterial genome acquired by the gut bacteria which carries the antibiotic resistance, or some kind of virulence that can be transferred from one bacteria to another. For example, E. coli LT or heat level toxin, which is similar to the cholera. So their origin is similar. So some point of time probably gene was transferred from the E. coli to cholera or the vice versa. So we have to. This will help you to understand how genes are transferred. There are some gene elements which are called jumping genes or transposons that we are going to discuss in the coming lectures. It will also help you to understand by utilizing the gene of interest. For example, spike protein of coronavirus. For a moment, let's not think about the bacteria, just about the genetics of the pathogen. So for example, if we are talking about the coronavirus, you want to make a vaccine out of this spike protein, then you need to take this spike protein gene and put it in a recombinant form in a bacteria or a vector, and that will express, and you can purify the protein and use it as a protein to inject human or animal or as a vaccine protein. It will also help you to understand the variation within bacterial species.
22.
What is a bacterial variation within bacterial species? That means that if you look at the whole genome, there might be some specific signature within the genome, particular sequences that may vary from one bacteria to another. For example, if we talk about again about say Neisseria meningitidis causing meningitis or Vibrio cholerae from Bangladesh, and you want to compare that Vibrio cholerae from say South America, for example say Peru. So these strains, if you do a sequencing and look at the genome, they will be different. So this will help you to understand variation with the bacterial species. Why does the bacteria undergo variation? Because they undergo variation because that helps them to escape the immune response. And lastly, it will also help you to understand the epidemiological background of an infection. For example, again, if you look at the spike protein or the coronavirus, now say that there may be multiple kind of types circulating all over the world. There may be these can be called clades or subtypes or types of the coronavirus. So how do you know that what are the different types? So you have to basically understand the genome of the virus. So this is what I'm trying to tell you, that when you talk about the genetics, it will help you to understand how this information can be utilized to your advantage. It will help you to understand about the disease process. It will help you to understand to undergo or prepare a vaccine. It will also help you to understand to develop newer antibiotic if the organism develops some kind of a resistance to different antibiotics. Okay, let's uh, let's talk about first. Let's go back to your class 12 class and understand what is the code of life. We have to understand the code of life lies in the DNA. It was in 1953 that you realized Watson and Crick first decoded the code of life, and they found that DNA is the material inside the cell that codes for all the proteins, and proteins are subsequently proteins in terms of enzymes. They will form the lipid or they will form the carbohydrate. So basis of life is hidden inside the DNA molecule. So this, what is the central dogma, or what is the central truth of biology, or like you say the central principle of biology, molecular biology, that the information is carried, genomic information or codes are encoded in the DNA. Then DNA is transcribed into messenger RNA, and this messenger RNA can be of carries the information to make synthesis of protein. So this mRNA is subsequently translated by the ribosome in the cytoplasm into different kind of protein. So this is a central dogma of biology. In principle, that DNA is converted into RNA, RNA is converted into protein, and protein is basically synthesized in the cytoplasm and through the ribosome, and this ribosome translates the messenger RNA into protein, and this protein finally forms the different component of the cell structure that we're going to discuss a little later. And about the RNA, we are going to discuss in very brief, because the idea of this today's lecture is not to go into details about the molecular biology, but to look at the bacterial genetics from the point of view of application into infectious disease. So in that way, today's lecture will be basically structured to understand our relationship in terms of infectious disease. What is a bacteria? Bacteria is basically prokaryotic cell. Now you have to understand that you already have understood the difference between the prokaryote and the eukaryote in your class 12 lecture, and we will realize the prokaryote is the earliest form of life once life evolved. So between the prokaryote and the eukaryotic cell, from the prokaryote to develop into eukaryotic cell, the evolutionary time was probably, the time taken for this development was probably 1 to 1.5 billions of years. That means if you just for a moment, if you compare the computer of the 1980s, so XT or AT computers, which is very slow and huge computers, and you compare the tablet of today's, that's the kind of difference we are talking about. If you look at tablet or if you look at desktop today, which is extremely sophisticated, and if you look back and say that at a computer from 1980s, and you say this is very rudimentary and it doesn't have this sophistication. So prokaryote is like the computer of the 1980s, where it functions as a life, but it is not as sophisticated as evolved as the eukaryotic cell. Let's quickly look at the difference between the prokaryote and eukaryote, just to revisit your class 12 lecture. A prokaryote has a rigid cell wall, bacterial cell wall. It also has a plasma membrane. But then inside, if you go from inside to outside, outside to inside, you have the rigid cell wall, then you have the plasma membrane, and inside you have the cytoplasm. And inside the cytoplasm, the DNA is present in the cytoplasm itself either in a linear form or in a compact form, depending on whether the bacteria is at the time of replication or it is just in a growth phase. The other thing you have to understand is there may be in bacteria there is only single copy of the chromosome. There is only one copy of the chromosome. That's why it is called haploid number of chromosome. So bacteria will always have a haploid chromosome. But it doesn't mean that it doesn't have extra chromosome. It might have some extra chromosome in the form of a plasmid or in the form of a say bacteriophage DNA. So extra chromosomal presence in the bacteria is possible, and invariably most of the bacteria will carry extra chromosomal gene in their cytoplasm. They may be a plasmid or they may be a bacteriophage, but they may have an independent or they have maybe cell cycle division along with the bacteria. If you look at the eukaryote, then you realize that there are a large number of membrane bound organelles. This can be rough endoplasmic reticulum, this can be mitochondria, this can be Golgi apparatus. These are basically structures which will form different kind of, they translate the protein, produce glycosylation, and process them in the Golgi apparatus and send it to different structures. Similarly, you have mitochondria in the eukaryotic cell. Mitochondria, you realize, a different kind of gene inside the mitochondria, and the eukaryotic cell will always have a diploid number of chromosomes. For example, if you talk about human cells, human somatic cell, then you have 22 plus XY, so you have 23 pair of chromosome in a eukaryote. Whereas, as I already told you, in the bacteria you have only single copy of the chromosome. That's why we say that the bacteria always has a haploid number of chromosome. Let's quickly look back at the bacterial cell wall, because we are going to talk about bacterial genome. If we look from the outside to the inside, you will see a large number of appendages outside. These are called pili. There is a big structure on the which is called flagella. This flagella is for movement of the bacteria. The small structured appendages on the surface of these are pili. These pili are composed of protein. Flagella is also composed of protein. So these are encoded in the genome of the bacterial genome or haploid chromosome. Inside the bacteria, if you go inside from the cell wall to the inside, then first you have the cell membrane or plasma membrane, then you go inside, you have the cytoplasm. So in case of bacteria, there are no membrane bound organelles. That means the bacterial DNA is not enclosed within a nucleus. So there is no nucleus, there is no nuclear membrane, there is no mitochondria, there is no rough endoplasmic reticulum, there is no Golgi apparatus. So everything is synthesized in the cytoplasm. The cytoplasm will have all the components necessary for production of the replication of the DNA as well as replication of the protein. So a large amount of ribosomes, which are the machinery that translate messenger RNA into protein. So the DNA of the bacteria gets duplicated, it forms the messenger RNA, and this messenger RNA are translated into protein right in the cytoplasm. So what we do need to understand? We need to understand about the cell wall structure of the bacteria. We need to understand a little bit about the cytoplasm. Cytoplasm, as you understand, that is the basic matrix of the cell where all the components survive. So it's a very dynamic kind of structure of the cell. Then as I told you, this is the chromosome. This is what we're going to talk about basically today. Ribosome are the structure, as you understand, that translates the messenger RNA, that is transcribed from the DNA into protein. And in addition to this, you need a large number of very specialized kind of enzymes. There are a huge number of different kind of enzymes in the cell. There are some housekeeping enzymes which are necessary to form housekeeping enzyme, etc. But the enzyme that we are talking about, these enzyme polymerases or restriction enzyme, these enzymes are necessary for the replication or duplication or multiplication of the bacteria inside the cell. So you have to understand that DNA polymerase or RNA polymerase are the enzyme that forms the that duplicates the DNA inside the cell. This is what we are going to talk about in a little while. You also have to understand that the chromosome of the bacteria inside the bacteria is not a constant rigid structure. What I am trying to tell you is that it is not that 100% one bacteria will be similar to the other bacteria. That means the bacteria, most of the bacteria will have the capacity to absorb genetic material or DNA or genetic material, fragments of the DNA from outside, from the environment, either through transformation or through infection of the bacteriophages that comes inside the cell, and this DNA may be integrated into the chromosome and it can be a part of the bacterial chromosome. So there is some kind of plasticity. What is the plasticity of the DNA? That means the bacterial DNA has the capacity to absorb DNA from outside, and this will give some evolutionary advantage to the bacteria. It may, for example, this additional DNA that comes from outside may give the property to the bacteria that it is able to produce now the pili on the surface of the cell, which was not present before the DNA came in. Similarly, it may help the bacteria produce large amount of labile toxin, heat labile toxin that we talked about as in cholera. It may help the organism to form a capsule. It may help the organism to produce certain kind of hemolysin, so on and so forth. So this plasticity of the prokaryotes is necessary for the bacteria to evolve to make it fit in the surrounding. As you understand Darwin's theory that only the best fit survives. So the bacteria also tries to preserve its DNA, but at the same time it will also acquire DNA from outside so that it becomes plastic. It has more varieties or more diversity, and it is much more capable of withstanding the environmental pressure or the pressure within the host immune system. Okay. So this is how a bacteria looks like. As you understand, you have a DNA. You have to understand there is no nucleus, only no nucleoli, and the ribosome, huge number of ribosomes are spread all over the cytoplasm. The moment the DNA is transcribed to messenger RNA, this ribosome will stick to the messenger RNA, will immediately start producing the polyproteins, and these polyproteins subsequently will get cleaved into bacterial protein, so on and so forth. Okay, this we already discussed. Let's start with the how do you realize that the DNA carries all the genetic material. This was realized in 1928 by Frederick Griffith in an experiment. What is the experiment about? He This is called the transforming principle or discovery of transformation. What is the transformation? That the bacteria is transformed into, from a non-pathogenic bacteria, it gets transformed into a pathogenic bacteria. How does that happen? So you have two strains of bacteria. One is smooth strain, which is virulent. It is indicated in the red capsulated bacteria. This is a pneumococcus. So Streptococcus pneumoniae, smooth strain, because it has a capsule, so it is extremely virulent. So if you take this strain, grow it in a culture plate and inject it in a mice, the mice dies because it's a virulent strain. So this smooth capsulated live strain of the bacteria will kill a mice. So extremely virulent. Similarly, same organism, there is a rough variant, doesn't have the capsule. So when you inject the non-capsulated Streptococcus pneumoniae, which is called rough strain, inject into the mice is able to withstand the bacterial strain inside and is able to the immune system of the mice, macrophages, antibodies will kill the bacteria and the bacteria survive. So you understand that there are two strains, the live strain, both are live strain. The smooth variety will kill the mice, whereas the raw variety doesn't. Now what you do is you heat kill the smooth strain that is virulent, and you take the live strain of the raw strain. So kill strain and the live raw strain are mixed together. So this is the killed smooth strain. It is not able to replicate because it is dead. Then you have the live strain, you mix up it in a test tube. You heat kill the bacteria, you take the solution in a test tube, heat it to 60 degrees centigrade for half an hour, the smooth strain of the bacteria will die. Then you mix up with the live raw strain, incubate for half an hour, mix them together and inject it to the mice. After 3-4 days the mice will die. What is the inference? Inference is that the RAF strain, non-virulent, non-capsulated RAF strain, has acquired something from the dead strain of the bacteria, and the RAF strain now, because it is the only one which is alive, it has acquired certain amount of substances from the dead virulent bacteria, and it has now become virulent capsulated strain. If you isolate the bacteria, you will see this smooth strain. How did this happen? This happened because the rough strain was transformed by something of from the heat killed smooth strain, and this transferable element was the DNA that we discovered in 1953. So in 1953, James Watson and Francis Crick, Watson and Crick, were awarded Nobel Prize because they have decoded the life. And what is the code of the life? That is in the form of the DNA. Let's quickly look at the what is a bacterial genome. I told you bacterial genome is basically a chromosome. Unlike the eukaryotic, where the genome is highly packed with the histones and other nucleoproteins. The bacteria doesn't have a histone protein, so but it is very tightly coiled and it is present in the cytoplasm as a loose kind of ring, just like a necklace. If you when you come for practical some point of time, if you kill the bacteria, suppose with some kind of a alkali or sodium hydroxide or maybe bile or something that will destroy the bacterial cell wall, you can pick up the DNA as a string from suppose you take the bacteria on a couple of colonies of bacteria on a glass slide, and add some kind of a detergent that will destroy the cell wall, and with a loop you should be able to pick up the string of the DNA. That's because this is loosely present inside the cytoplasm, just like a necklace, but it is highly coil structure. It's a duplex DNA, extremely tightly coiled, but unlike the eukaryotic DNA, this DNA of the bacteria haploid chromosome doesn't have histone, but it's a highly coiled structure. As I told you, in addition to this structure, the chromosome, host chromosome of the bacteria, in addition you can have additional extra genetic material in the cytoplasm in the form of the plasmid. This we are going to talk about little bit about plasmid, but very briefly, plasmids are circular extrachromosomal DNA may be present in the cytoplasm that may autonomously replicate, that has got nothing to do with the host chromosome replication. So, a plasmid is an extrachromosomal DNA that is present in addition to the host chromosome, and it has the ability to autonomously replicate inside the cytoplasm. So, if it replicates from one copy, it becomes two copies, and the second copies can be transferred to a different bacteria through different mechanism we are going to discuss. What is the advantage or disadvantage to this bacteria? That if the bacteria acquires a plasmid which has additional property, like for example drug resistance, antibiotic resistance, then the bacteria will acquire antibiotic resistance. This is exactly how the hospital infection is carried through. We are going to discuss about the jumping genes or genes that can transfer from one bacteria to another in a little while. Okay. Let's quickly look at the structure of the DNA. You already understand there are two types of nucleic acid inside the bacteria. In viruses, you know that viruses can either have DNA, because viruses are extremely primitive, so they can only have DNA or they can have RNA. They cannot have both. So either the DNA virus can be, say herpes virus, it will have only DNA, or it can be a polio virus, which can, just for an example, it can have HIV, which will have only RNA virus, or hepatitis, who can only be the DNA virus. But in bacteria, because bacteria is virus is a non-living structure, it only comes alive inside the cell, so it uses the cellular mechanism to replicate. Therefore viruses can exist either as a DNA or RNA, but rest of the structure required for replication or survival of the viruses, it acquires from the inside the eukaryotic cell or a human cell or animal cell, whereas the bacteria can have an existence outside the cell. Therefore it has the ability, it has all the structure that are required to make progeny, its own subsequent generation. So it will have all the component of a cell. It will have the DNA, it will also have RNA. What is DNA? DNA and RNA basic difference is that the sugar is different. In DNA you have deoxyribonucleic acid, RNA you have ribonucleic acid. As I said, in bacteria the role of the RNA is basically to in the formation of proteins, because DNA, as I told you in the central dogma, that DNA is transcribed into messenger RNA or tRNA or hRNA. So and finally the messenger RNA is translated into protein. Let's quickly look at the basic building block of the DNA, or deoxyribonucleic acid. So in a DNA basically you have three main groups: a phosphate group, which comes from the nucleotide triphosphate, a pentose sugar, either it can be deoxyribose or it can be ribose sugar, and you have a nitrogen base. So these are the three main components of a nucleotide, basic building block of the DNA. So a nucleotide will have a phosphate group, it will have a sugar, and it will have a nitrogen base. This nitrogen base can be a purine, adenine and guanine, or it can be nitrogen can be pyrimidine, which is cytosine or thymine. Okay, so we're talking about the basic building block of the DNA is a nucleotide. What is a nucleotide? Is basically composed of a phosphate group, it is composed of a sugar, and it is composed of a nitrogen base. This nitrogen base can be either purine, adenine and guanine, or it can be a pyrimidine, which is cytosine and thymine. So these four structure that you have, four nitrogen bases that you have, these are the building blocks of the. So you have adenine, adenosine triphosphate, thymine triphosphate, cytosine triphosphate, and guanosine triphosphate. So ATP, TTP, CTP, GTP, this is the building block of the DNA. This is what makes the DNA strand. Okay, so this is how the structure looks like. So you have a base sugar, either it can be this can be either a purine, or this is a pyrimidine. This is a pyrimidine, and then you have a ribose sugar, and then you can have a phosphate molecule here. What you have to understand is that the nucleotide triphosphate will have a preceding nucleotide triphosphate here and a succeeding nucleotide triphosphate here. The preceding nucleoside triphosphate will bind to the incoming nucleotide triphosphate at the 3' position with the hydroxyl ion. So this hydroxyl group is important because this is where the phosphate molecule will come and bind. So this phosphate molecule will come and nucleotide triphosphate molecule of the coming nucleotide triphosphate will come and bind to this. So this binding is very important. Then you realize that one nucleotide triphosphate in the DNA strand always complements with another nucleotide triphosphate on the other strand. This is called complementation. This complementation is between A and T, adenine and thymine, one purine and the other pyrimidine. So it is never between purine and purine or pyrimidine and pyrimidine. So you will have adenine with thymine, or you have cytosine with guanine. Now you have to understand that the backbone of the DNA strand is basically parallel strand of anti-parallel two strands made up of sugar phosphate backbone. This is the sugar, this is the phosphate, and on the inner component of the DNA strand you have the nitrogen base. So one nitrogen base will always complement with the other nitrogen base, and this is paired through a hydrogen bond. Okay, so a DNA strand basically is two strands of DNA, and these two strands of DNA is formed because of the complementation between A and T and G and C. Now we have to understand that the binding of these two is through hydrogen bond, and this binding hydrogen bond is in the, if you have G and C, guanine and cytosine, then there will be 3 bonds here, whereas in the A and T there will be 2 bonds. What is the meaning of this? The meaning of this is that if you have higher number of G plus C content in the DNA strand, then the binding strength will be, these two bond will be very high. So that if you are doing a PCR and you want to separate these two strands of DNA for annealing of primers, then you need the higher temperature you need to raise the temperature to higher, so that the number of, it has to break the higher number of bonds between G and C. So G and C had 3 hydrogen bond, whereas A and T has 2 hydrogen bond, and this is why it is necessary, the higher the G plus C content of the DNA strand, higher is the melting temperature. So here the TM, when you design a PCR, you are normally, you do not choose an area which is extremely high G plus C content, because in that situation your temperatures can be raised need to be raised higher than 94 degrees centigrade. Usually 92-94 degrees centigrade is taken as the melting temperature. So this is what I was talking about, that the DNA strands are basically a strand which is composed of a repeats of nucleotide triphosphate with sugar phosphate backbone and the nitrogen base on the inner side. The backbone is made of sugar phosphate and complementation of the nitrogen base on the inner component, and these are anti-parallel. Uh, as I told you, this hydroxyl group at the 3' end of this sugar is very important, because this hydroxyl group will form the phosphate bond. This is a extremely energy rich bond which requires a large amount of energy to break it. So this phosphodiester bond is a covalent bond, and this is how these strands are attached to. So this is not easy to separate these two strands, two nucleotide from each other, but if you apply heat, these two strands will separate, but this will never break. So for this breaking, you need a special enzyme which is called restriction enzyme. This restriction enzyme has the ability to cut this covalent bond, that we are going to talk about in a little while. So how does the bacteria look like? Prokaryote look like, as I told you, it has the cytoplasm, and in the cytoplasm we have all the ribosomes that are necessary to translate the messenger RNA into proteins. And when the bacteria divides, there is something in prokaryote. In the eukaryote you have the centromeres where the replication of the chromosome starts, but in bacteria a segment from the cell wall which is called mesosome invades into the cytoplasm, and it separates the cytoplasm, and it induces the proliferation or replication of the DNA molecule. That's what we're going to talk in a little while. Talking about replication or division of the bacteria, this is a binary division, as you already know. From 1 to it becomes 2, from 2 to it becomes 4, from 4 it becomes 8, from 8 it becomes 16, and so on and so forth. So what is the doubling time? Doubling time is basically when it becomes from 1 to 2. For E. coli it takes about 20 minutes. For Mycobacterium tuberculosis, which is a slow growing organism, and you will realize that for you to grow the E. coli in the laboratory, it takes about 18 to 24 hours to get a full plate of bacteria. Whereas for Mycobacteria, it takes 24 hours to divide from 1 to 2. So it takes about 6 to 8 weeks for you to get a culture positivity in Mycobacteria in a normal conventional culture. Whereas in Trypanosoma pallidum, or that is causes syphilis, the replication time is even higher. So incubation, what I am trying to tell you is that the whole duplication time is directly related to the incubation of the disease. If you talk about E. coli, when it causes urinary tract infection, when the E. coli invades into a individual, male or female, causing urinary tract infection, the incubation period for the disease may be only 2 to 3 days. Whereas for Mycobacterium tuberculosis, it may be several weeks. Whereas Trypanosoma pallidum, it can be anywhere between 9 to 90 days. So it is extremely similarly Mycobacterium leprae. It take months before the organism actually makes its presence felt in terms of producing the clinical picture, because if the bacteria is able to multiply rapidly like typhoid, it will produce disease very fast. If it is a Vibrio cholerae, it multiplies very fast. Within 4 or 5 days the patient will have severe symptoms. But for example, Mycobacterium leprae, Mycobacterium tuberculosis, it will take long time before the symptoms. So doubling time is important to understand, because it is directly related to incubation period of the infection. Let's quickly look at how does a DNA replicate in the bacteria. A bacteria has something called origin of initiation in the chromosome. There is a particular sequence in the bacteria which is called ORI, or origin of initiation. So there is a, I'm not going to go into detail, there are certain sequences in the DNA that is recognized as the replication initiation. So at this point the replication will all start. How does the replication start? First the DNA will unwind, and because as I told you, this is a very highly coil structure. It is highly strong positive strand coil structure. So this has to first unwind. The chromosome has to unwind. It has to become loose. Once it becomes loose, then all the supercoils are gone. It becomes a simple linear strand, and this linear strand, the two template has to be separated, and these two template separation is being done by an enzyme called DNA helicase. It's a part of the DNA polymerase system. This is enzyme unwinds the separate, it keeps on pushing the separation along the strands of the DNA, and this helicase will keep on pushing the separate, and these two strands will slowly keep on separating, and once these two strands keep separating, this becomes a linear strand, this becomes a linear strand. So one of the linear strand is called, this is what I was talking about, that ORI, origin of initiation. So in origin of initiation, the two strands will separate, and the replication will start. Then the replication, always, as I told you, the helicase will separate the DNA strand into a linear strand, and then these two strands of the original DNA becomes two linear single strand that can act as a template for the synthesis of new DNA strands or daughter DNA strands. So one of the DNA strand that is transcribed from 5 to 3 prime, this is the 5 prime to 3 prime. You understand that this is the 3 prime to, this is 5 prime, this is 3 prime, this is 5 prime. So in there are two strands, the leading strand and the lagging strand. In the leading strand, as the helicase pushes the DNA strand into separation, in the leading strand, the DNA strand will be synthesized by DNA polymerase III in a linear continuous fashion. As the strands keep opening up, the DNA polymerase will keep on adding nucleotide triphosphate, and there will be continuous increase in the length of the DNA, and the new strands of DNA will form. But in the lagging strand, what happens is, in the lagging strand, the lagging strand of the DNA template, the synthesis, it cannot be in a linear continuous fashion. So what does it do? The DNA polymerase adds a RNA. As it opens up, it will form a RNA primer. RNA primase is an enzyme that forms small RNA primer. These RNA primers are necessary to form DNA, because DNA polymerase de novo cannot start a DNA synthesis. So RNA primer or RNA primer is necessary to form the template. So the RNA primer will complement to the lagging strand, and then the DNA polymerase will keep on adding nucleotide triphosphate, and the helicase will push further up, and a new space will be created, and then another primer will be, so the lagging strand will be synthesized in a short fragments, small, as it keeps opening up, small fragments of the DNA will be synthesized. These fragments are called Okazaki fragments, and these fragments are finally stitched together by removing the RNA primer by the DNA polymerase 1 enzyme, polymerase 1. DNA polymerase enzyme 1 has the exonuclease activity, that it removes the RNA primer and it can repair. So at some point of time, this discontinuous fragments of DNA that are used from synthesized from the lagging strand will be a continuous strand. So this is how this, and then you have to understand the three important proteins that are necessary for the synthesis of the DNA is DNA polymerase 3. This DNA polymerase, in a test tube, when we make the PCR, when we do a PCR or when we do a RT-PCR, real-time PCR, this DNA polymerase is used for, in the test tube, you can separate the two strands. Unlike the inside the cell, one in the test tube, the DNA strand are absolutely linear. So there is no, you do not require a helicase enzyme. So DNA polymerase is in the form of Taq polymerase. Taq polymerase is called Thermus aquaticus. So this Taq polymerase or DNA polymerase that has been synthesized, this DNA polymerase we are talking about, for example, E. coli. E. coli DNA polymerase cannot withstand temperature up to 94 degree, which is an important step in the PCR or RT-PCR. So you need to utilize a heat resistant DNA polymerase, which is in the form of Taq polymerase. So Taq polymerase are enzymes which are acquired from in a bacteria which are found in the hot spring, and this bacteria can withstand temperature up to 98 degree. So DNA polymerase from the heat resistant bacteria are called Taq polymerase, and Taq polymerase are the enzyme that are required for synthesizing DNA in a test tube, that is in the PCR. The other enzyme that you have to understand is called helicase. As I told you, helicase will keep separating the DNA strand. Topoisomerase is another important enzyme that is necessary for bacterial replication, DNA replication. The topoisomerase unwinds the coil structure of the DNA. So it will cut a nick into the one of the DNA strand, and then DNA strand will become a negative strand. Negative strand in the sense that the supercoiling is removed, then it will become two complementary strands. So the function of the topoisomerase is the uncoiling of the DNA. The function of the helicase is to separate the two DNA strands into single strand. Now we have to understand that this helicase enzyme has been targeted as an antibacterial antibiotic. So you have fluoroquinolone group of drug, for example, ciprofloxacin or other fluoroquine, levofloxacin, that specifically helicase is an enzyme that is a part of the DNA gyrase. So helicase, so the DNA, the function of this DNA gyrase or helicase is to separate the two strands of the DNA. An antibiotic like ciprofloxacin or levofloxacin will block the function of this DNA gyrase or helicase, and the DNA strands separation will not be possible. Therefore, the bacteria will not be able to multiply. So ciprofloxacin, as you have heard, is used for most of the common bacterial infection and pneumococcus, salmonella, at least in the beginning. So but the bacteria are very smart, so they will introduce changes in the DNA gyrase or helicase, and it will become resistant to antibiotic. So what I might as well tell you at this point of time. So when you synthesize this DNA, not inside the cell but outside the cell in a test tube like polymerase chain reaction, so what are the things that you need? You need the template DNA from the bacteria of interest or virus of interest. Then you need a primer that you need to bind, because in the test tube you will not have other system, you will not have the RNA. So you need the primer. So you need the template DNA, you need the primers, and you also need the DNA polymerase. And in addition to this, you need the nucleotide triphosphate DNTPs, and you need magnesium. So you have the, what are the components? You need the template DNA, you need the DNA polymerase, you need the RNA primers, and you need the nucleotide DNTPs or deoxyribonucleotide triphosphate to make the new strands of DNA. Along with that, you will require some elements like magnesium to stabilize the whole procedure. So in fact, this replication of the DNA or duplication of DNA in the cell has been reproduced in the test tube using exactly the same component: DNA polymerase, primers, DNTPs, magnesium, template DNA, and this is what we call is polymerase chain reaction in the test tube or real time polymerase chain reaction in the test tube. So this is what I was talking about. These are DNTPs, and these two templates are, so DNA synthesis always is from the 5 prime to 3 prime. So the 5 prime to 3 prime is continuous, as it opens it will keep on lengthening, whereas in the lagging strand you have the fragmented DNA Okazaki fragments, and the DNTP comes in through the hydroxyl ion and new strands are formed. This is continuous. 5 to 3 prime is continuous, this is the leading strand, and this is 5 to 3 prime, this is a lagging strand. So this is where the Okazaki fragments are formed. So leading strand 5 to 3 is continuous, whereas the other one it is a fragmented strand. Okay, I think this is as much as we need to know about the bacterial replication. Very quickly, this is just the repeat of the thing that I am going to discuss about. This is what I was telling you, that DNA is replicated into RNA, transcribed into RNA. You have different kind of RNA. You have messenger RNA, you have tRNA. Messenger RNA is the one which encodes all the protein molecules in the nucleic acid. tRNA is a clover leaf structure. It carries an anticodon at one end of the loop. At the other end, it carries the amino acid. So tRNA is basically the carrier of the amino acid, forming an codon. This is anticodon, and this anticodon will bind to that particular, and this anticodon will complement the codon on the messenger RNA, and the protein will be synthesized. So this is in a nutshell how the protein is synthesized in the what is the function of the ribosomal RNA? Ribosomal RNA basically stabilizes the messenger RNA inside the ribosome. So, and we have to understand that this ribosome is where the messenger RNA is translated along with the tRNA with the help of the rRNA into proteins. So therefore, the ribosomes are the target for a large number of different antibacterial antibiotics. For example, chloramphenicol, for example, aminoglycosides, for example, macrolides like erythromycin, chloramphenicol, tetracycline, gentamicin. All of these work on blocks the ribosome, but the organisms are very smart because they will also change the component or amino acid mutations in the genome, and they will produce a different form of ribosomes so that the antibiotic may not work. This is how the drug resistance occurs. This is the what we are talking about, the central dogma. DNA is converted into RNA, messenger RNA. Messenger RNA along with the tRNA is converted into a protein. So replication is transcription, the DNA into messenger RNA translated into different protein. As you understand, this is what I was talking about. The tRNA at one end it carries the protein amino acid. At the terminal lobe it is a clover leaf structure, and at the other end you have the anticodon. So codon anticodon will, this is the messenger RNA, codon anticodon matching happens. This whole lining up occurs in the ribosome, and then the subsequent it moves to another step, and the subsequent tRNA comes in, and then the chain of protein continues. Now we have to understand how this code takes place. You already know that there are only 4 bases nucleotide triphosphate A, T, G, C. So if you, and then the genetic code is always in the form of 3 bases code. So 3 nucleotide in a tandem forms a code of protein. So you may have AAT, CCC, GGG. So the 3 nucleotides form a codon. As I already told you that codon anticodon there is only 3 pairs. So these this pair of these 3 codon will complement the anticodon and the tRNA, and this tRNA will take a particular. So based on this particular anticodon or this codon, the pairing will take place. So this is always very specific. There is not much possibility of any error here. So what happens is there are 4 nucleotide triphosphate and 3 forms a codon. So a combination of about 64 combination can take place. Out of this 64 combination of codons, 3 codons UAA, UAG and UGA basically form stop codon. This is where the these codons are converted into codons. These stop codons are present, as you understand, through these are in the messenger RNA. This is in the RNA. So this is where the ribosome will stop and the protein will not be synthesized. And this is AUG is the initiating codon. So out of the 64 codons, you have 61 actually codes for amino acid and 3 termination code, one initiation code. Let's quickly look at this. This is the messenger RNA. This is the tRNA that comes in. Codon anticodon complementation takes place, and then the subsequent tRNA comes in, and you have a elongated protein that keeps on elongating, and this is how these proteins are synthesized, and messenger RNA is translated in the ribo, and this whole thing takes place in the cytoplasm. Let's quickly look how as I told you in the beginning, that the DNA in a bacteria is plastic. That means it has the ability to acquire or lose or cause mutation changes in the DNA. So it's a plastic, it has a much more plasticity. So what are the plasticity? Plasticity can be in the form of mutation, or plasticity can be in the form of acquisition of larger chunks of longer stretch of DNA. So these mutations are normally defined as a single point mutation, deletion, insertion, etc., which can be helpful, it can be lethal, it can be silent, sometimes it can be nonsense. So what happens if you change a particular sequence nucleotide in the chain of DNA, in the chain of DNA. Suppose this is A is replaced with the G, then it's from the glycine it becomes an arginine. So this is a mesh sense. It causes replacement of amino acid. How does it? Sometimes it will affect the function. If it affects the function, then the bacteria may die, or it may cause a different kind of inefficient or highly efficient enzymic activity. Nonsense. The termination. If suppose CA is converted into UAA, this is a termination codon. So what happens is the ribosome stops and the protein is, you have a truncated protein. Similarly, if you put in a new nucleotide somewhere inside or delete this nucleotide, then the whole frame of the reading frame is changed. That means you normally read 3, then 3. If you remove this, then the reading frame will change, so it will form a different kind of amino acid, different kind of protein, and which may be functionally different, which may be helpful, which may be detrimental. So when you're talking about mutation, normally it says that you only change, the bacteria only changes only one. So it can be substitution, or it can be an insertion, or it can be a deletion. Substitution is replacing adenine with guanine or cytosine with thymine. Insertion, you putting another nucleotide outside. So this will also some kind of a frame shift mutation. This will also cause frame shift mutation. Normally DNA polymerase has, if this happens in the DNA, this we are talking at this moment, you are talking with on the RNA, because you see this is that there is a uracil. So when this happens in the DNA, mutation happens in the DNA polymerase has the capacity to do a proofreading. So it has a exonuclease activity, so it can remove this particular substituted nucleus with the appropriate nucleus. So this will be corrected. But the RNA polymerase doesn't have that. So the RNA polymerase, the mutations will be present in the. So these mutations are mutation that can happen in the DNA, then it can get converted into the RNA molecule. Let's look at what are the different ways the DNA of bacteria can change. So the bacteria can acquire DNA from the surrounding. This is called transformation. Suppose, for example, in the intestine, if you have a large number of different bacteria. Suppose there is E. coli, there is Salmonella. The E. coli dies and its cell wall breaks. The DNA is present in the environment. Then the Salmonella picks up the DNA from the E. coli, and the E. coli DNA becomes a part of the Salmonella DNA. This is called transformation. Conjugation. What is a conjugation? So, conjugation is basically meeting of two different individuals. So what happens is that it's like having a sex between male and female. So one bacteria we have a pili which could connect to the second bacteria. This is called a sex pili, and the genetic material is transferred. Sometimes a DNA is transferred to a bacteria by a virus. This I am going to show in a little while. So this is how the transaction looks place. This is the bacterial DNA, this is the virus infects the bacteria, then the viral DNA is inserted, and this bacteria, the bacterial virus DNA is replicated, and the virus destroys the bacterial DNA, and this bacterial DNA, when the new virus forms, a part of the bacterial DNA is picked up by the virus, and when this virus infects a second bacteria, it also gives a part of the first bacteria to the second bacteria. So this is called a transaction. For example, one of the important transaction phenomena that we know is the Corynebacterium diphtheria, where the Corynebacterium diphtheria acquires the toxin production, diphtheria toxin, or cholera, Vibrio cholerae, where it produces the cholera toxin through transaction. Similar transformation, I already told you that the bacteria dies and picks up the DNA, and then the new bacteria becomes acquires the part of the. There's something. This is what sex pili conjugation looks like. Through the sex pili, the one of the bacteria, donor bacteria, can transfer a large amount of DNA into the second bacteria. This is how it looks like. This is sex pili. The plasmid, the autonomously replicating, it replicates, and a fragment, a part of the DNA, a copy of the DNA is given to the next one, and it transfers whatever properties they carried by plasmid, drug resistance, etc. There are something called jumping gene. There are something called jumping genes. What are these jumping genes? These are called mobile genetic elements. For example, there is a small fragment of insertion sequence. This is called transposable genetic element. The smallest type of insertion elements are maybe couple of sequences, couple of nucleotide triphosphate bases. These are called insertion sequences, transposons, because they have ability to transpose from one segment to another. It has the enzyme that will cut the DNA at one end, and when it jumps to a second one, it will have the ability to. So what happens is, suppose this is the insertion sequence. This is the insertion sequences. These insertion sequences carry enzyme which are called transposes. Transposes has two components, restriction and ligase. So it will cut the DNA and it will ligate. So the whole chunk of this small jumping gene will get inserted to different, and when it jumps from one side to another, from one bacteria to another, it may carry drug resistant genes, it may carry pili producing capacity, it may carry toxin production and given to a different bacteria. So this is a transposon. It can jump from a different bacteria. When it jumps to a different bacteria, this jumping element can be recognized because it has a very specific sequences, and these specific sequences can be, because you know the sequence of the transposon at both the ends, it is flanked by the insertion sequences. So these insertion sequences are very characteristic. So you can always, there will be inverted repeats at both the ends. The sequences are inverted. So by looking at the insertion sequences or inverted sequences, one can identify these transposable elements. One of the largest, Barbara Macklin got the Nobel Prize for her work in the jumping genetic elements, transposons, which are actually very important as far as the drug resistance is concerned. So I already told you that one of the jumping element was plasmid. Plasmids are extrachromosomal DNA which has the ability to autonomously replicate in the bacteria, and it transfers a large number of different properties, phenotypic properties, fertility, bacterio cyanogens that forms different kind of fluorescent pigment. It can transfer resistance, antibiotic resistance. It can transfer bile production. It can produce proteases, so on and so forth. So plasmid sometimes can be transferred from one bacteria, mostly common in case of hospital acquired infection, because in hospital a large amount of antibiotic resistance spread to all over the hospital through plasmid in a very short period of time. But as I told you, the jumping elements, so we know that there are different kind of jumping element, include something called a transposable elements, pathogenicity island. What are the pathogenicity island? Whereas the insertion sequences are very small, transposable pathogenicity island very large. If the transposable elements are insertion sequences or transposons are suppose 10 nucleotide bases long, or say 15 or 20 nucleotide bases long, the pathogenicity island can be 150,000 long base, and these are fragments of DNA that are present in the bacteria. When it dies, the whole chunk of DNA is taken up by a second bacteria, and this second will acquire the pathogenicity island. How do you recognize the pathogenicity island in the genome? In the new host chromosome, their G plus C content is different. They will be always flanked by the insertion sequences, inverted sequences, and wherever the pathogenicity island is inserted, the target DNA is duplicated. So these are some of the signs that you can identify the pathogenicity island. If the insertion sequences are present in the DNA, this will be always be able to jump from one element to another, or even within the chromosome it can change its position. But if these insertion sequences are lost, then this will become a permanent fixture in the bacterial chromosome, because it will not have the ability to jump from the particular position, because these insertion sequences with restriction enzyme and the ligase is not present. Okay. So a bacteria can acquire or lose its part of the genome through deletion event, or through horizontal gene transfer. It can acquire plasmid, it can acquire bacteriophages, so the plasticity can be increased, or it can through mutations or rearrangement it can form a newer form of variation within the so we are talking about jumping elements. So jumping elements, we know that it can be through transformation. Pathogenicity island can be through transformation, that is picked up by the bacteria from the environment. It can be through plasmid that can jump from one bacteria to sex pili or conjugation, or it can be through bacteriophages, like through which process is called transduction. Lastly, this is how it does: plasmid from one to another, transformation, pathogenicity island, fragments are picked up by second one, transduction by bacteriophages, conjugation by sex pili. You're going to skip this part. There are certain repeats of sequences, the jumping elements. This insertion sequence is repeated many times, or this can be in tandem. This can be in tandem. Can be in the tandem repeats, that it is in a continuous serial sequential manner. The sequences are present. These are some identification point of the bacteria. Lastly, we are going to talk about a very unique kind of system that is present in the bacteria. This is called CRISPR. What is a CRISPR? These are clustered, regularly interspersed, short palindromic repeats. What does it mean? Over a period of time, what has happened is the bacteria are protected against invading bacteriophages DNA. So what it does, so this is a kind of adaptive genetic element in the bacteria, CRISPR, clustered, regularly interspaced palindromic repeats. So what it does is, once a bacteria comes in, it infects, a part of the bacteriophage DNA, viral DNA, is archived into the CRISPR array. These colored bits that you have seen, these are fragments from past infection. These are fragments of DNA from past bacteriophage infection, and this CRISPR-Cas9 system of the bacteria has archived all the past infection DNA. So whenever an infection takes place with the bacteriophage and it is similar to a past infection, what it does is this CRISPR-Cas9 system, this is a gene editing system, will synthesize something called pre-CRISPR RNA or CRRNA, CRISPR RNA. These are CRISPR RNA, CRISPR RNA is something like messenger RNA. This is DNA, this is messenger RNA. So this CRISPR RNA will go and bind, target the specific site at the bacteriophage, and once there is complementation binding takes place, then the Cas9 gene or enzyme system will cut the DNA, bacteriophage DNA into fragments, and the bacteriophage will be killed. This system is called gene editing system, and this CRISPR-Cas9 gene editing system has been utilized in the molecular biology recombinant technology for editing gene with false kind of missense or nonsense mutations that can be harmful. So this is a gene editing system, but for bacteria this system is very innovative. It's an adaptive genetic element which helps the bacteria to survive by destroying a invading DNA. So in summary, what we discussed about is we talked about basic composition of DNA. We talked a little bit about the bacterial DNA. We looked at how the protein synthesized. We also talked about how mutations and gene transfer takes place. And we talked about little bit about how the genetic elements are transferred, how the plasticity of the bacteria acquired by acquiring transposon plasmid, and all these jumping elements are important because they confer a huge number of different properties which might be additional for the bacteria to survive in a competitive environment. Thank you.